Well, thank you again, Mustafa, for taking time. We're really excited to talk to you. And I think your work on cancer and bioelectricity is really fascinating, which would love to chat about, but maybe taking a step back, why don't you tell us a bit about your background and how you even got interested in bioelectricity? I believe it was when you were a kid playing with radios and getting electric shocks, but would
That's
love to
it,
hear the
huh?
story.
Yeah, that story has been around a while. It indeed started when I was about 17 and I built a radio transmitter from old valves and things. This is where I was born and grew up in Cyprus. And the thing is, I built this kit on my mother's mincing board. It had a nice shiny surface, but everything was out in the open. So we were dealing with live electricity. And my God, I got the electric shock several times. And I can tell you, I don't know if you or Mike, you must have had the old shock, Mike, in your times.
Many times.
Yeah, I mean, I was absolutely fascinated, and also scared by this force going through your body that could kill you. And after several times, I decided to turn it into ultimately a profession. It did work, by the way. I haven't turned it on for a while, but I was transmitting in the neighborhood. It was a crazy time. It was meant to work with just a Morse key, da, 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 da. And then I found a microphone, connected to microphone, and it worked with voice as well. Oh, it was incredible. And yeah, that was it. And I did, at some point after I became, I had my own lab and started recruiting graduate students and things. I did on a couple of occasions said to my incoming students, I said, look, if you really want to understand what bioelectricity is, you should go and put your finger in an electric socket. And then you will know what your PhD is all about. Well, this didn't go down too well at the time. So, but this has stayed with me. I mean, it's been a fascinating journey. It really is. And I'm sure same for Mike. We live with this something between a dream and a reality. In cancer, we do believe we have generated a whole new vision of the cancer process, in fact, the metastatic process, and all because of bioelectricity, which of course not many clinicians, with all respect, quite understand. This was one of the early problems, when I started sending papers to Nature and things, and they just couldn't hack it. The biologists couldn't hack the electricity or the bit of physics and things. The point came when I had to almost educate oncologists from bottom up, go down to basics and tell them about ion channels and remind them of Hodgkin and Huxley and all these things. And the rest is history. Here we are, running a journal on bioelectricity. The field has established all around the world. It's going through changes, developments, novelties all the time. Even today, just talking with Mike a minute ago, there's a paper in one of the cell journals talking about action potentials in glioblastomas. So every cancer seems, not every cancer, it is aggressive cancers are electrically excited. And the big question now is, what is this activity? What are these signals doing in driving the cancer process? We're beginning to form some ideas. So very exciting times.
Mike, go ahead. Sounds like you have a question.
Just still before we get into all the, I'm just curious, background-wise, what would you say in all of the courses, classes, all the education that you've had until now, what would you say were some of the most useful or the most essential things that have helped you make? I mean, you've made breakthroughs in this field. You're a leading figure in the bioelectricity of cancer in the modern times. Like, people ask me a lot, young people ask me a lot, what should I be studying? And obviously I have my own opinions, but I'd love to know from you, what did you feel was the most important for you?
Well, I have always followed my instinct. I get that sort of question all the time, and I always say, follow your instinct. Do what it is that you want, just like I myself did. And I did go on record once saying I could have been a road sweeper somewhere. My roads would have been the cleanest in the neighborhood. So I think, do what it is that you want to do, but do it well. Now, of course, with cancer, you're dealing with a deadly serious situation. So there is no leeway. There is no chance to get it wrong. And this I realized very early on because we made the initial discoveries on prostate cancer. And many of my friends, including some from the West Coast, said, Mustafa, what you should do is to set up a whole series of rigs down this corridor and blast through all the cancers. And I thought, yeah, this is a good idea, kind of world domination type thing. Then, when I tried, I got a very firm message back from the British establishment saying, do not double around. Just stay where you are and dig deep. This is not a kind of, a party game. Don't spread too thinly. And we kept on prostate until curiosity again had the better of us. And with one of my students, I go at breast. As you know, prostate and breast are share many properties, and so do their cancers. And so one, weekend we did some recordings from some breast cancer cells, and there it was, the voltage-gated sodium channel. And one of the best things I did was to nip up to Newcastle in the north of England, made a presentation to the Physiological Society. Good news about PhysSoc is the abstracts of the presentations are published. So we had that abstract published. And then the following year, a friendly group published a full paper reporting the same phenomenon in breast. And then from there on, it grew. So we now have, different people working on different cancers. Very interestingly, I mean, different nationalities seem to have picked up different cancers. So interestingly, for example, in Mexico, they were the ones who did cervical cancer. So it turns out that cervical cancer is quite common in Mexico. So those guys focused on that. And as you know, the standard way of diagnosing cervical cancer is with a smear test. So you just run a slide and collect some cancer cells. So they did just that, and then put the electrodes on top, and there it was, the sodium channel directly in human cancer cells. Japanese looked at stomach gastric cancer, again, because there's a tendency for cases of stomach cancer to be high in Japan, and there it was again. And the rest is history. I mean, the days have gone now when we would get excited, oh, another channel, another cancer, and just forget it. These channels are everywhere. Cancer is like brain. All the channels are there, doing their own thing, just like channels do in the nervous system, and not, of course, on their own. They huddle together, they form networks, and drive the very, very complex process of cancer. I say cancer, but we work mainly on metastatic disease, on metastasis, which is, of course, the main cause of death from cancer.
Before we start going into some of those details, I'm still curious about one of your background questions. Mm-hmm. So at the age of 17, when you said you got these electric shocks, what was it that you thought of electricity back then? And then also, what was the progression from at 17-year-old playing with radios, getting electric shocks, to starting your own lab? What was that progression to ending up in bioelectricity with your own lab?
Okay, well, that's a very good question, Aastha. The natural thing for me to do then was to study physics. So I did my university exams, some of them in Cyprus, finished them off in the UK at boarding school, and I entered Imperial College to study physics. One of the best things I did, actually, because what physics taught me is to understand the physical universe. And that's what physics does. So everything from black holes to galaxies to quantum mechanics and of course electricity and so on. And that was very, and physics would tell you or explain all these things to you with equations, not just qualitative phenomena. And this was very satisfying. The fact that you could write an equation and predict the outcome of your experiment and so on, and show that light is wave or light is a particle, depending on how you look at it and so on. So that was my first step up from those electric shocks into into turning what was a hobby towards a professional training. And then when it came to a PhD, I mean, I had some odds against me studying in the UK and so on as a foreign student at the time. So biophysics was just coming in. I should say, of course, that I'm now talking of the 70s on the back of the golden era of physics in the first half of the 20th century. I mean, these gods of again, quantum mechanics and relativity and uncertainty and all these things. I mean, it was incredibly satisfying. You really went home every day very happy almost. But I worked very, very hard. So biophysics, bioelectricity, the natural place to then settle was, of course, neuroscience. So I worked on several neural systems, but I was always driven by model systems. I think Mike does this as well. One of my scientific heroes are Hodgkin and Huxley, and what the giant axon of this squid did to our understanding of nerve excitation and conduction and so on. Now, they couldn't have done half those experiments if it wasn't for that model system. And I thought that was very essential. To understand complex phenomena, you really need to use appropriate kind of model systems and so on. So for me, at that time, obviously I didn't have an infinite choice. The retina was the model, and it's a beautiful system. So you could peel it from the back of the eye, and I used mainly fish at that time, and it would do this natural brain slice would survive in simple oxygen. You put the electrodes in. And there's a type of cell in the retina called a horizontal cell, which can code color. So now remember now, I hadn't yet seen a resting potential in any nerve cell or muscle. So because you're poking the electrode into the retina in the dark and flashing light, you knew you entered a cell because you would get some kind of a drop, and then the cell would respond to light. And one of the cells, a horizontal cell, would hyperpolarize to green light and depolarize to red. And I was fascinated by this, damn it. There is a cell, one synapse beyond photoreceptors. And it's already telling you milk is white, milk is black. Memory potential is going in opposite directions depending on the color of the stimulus. And I mean, this was incredibly powerful signaling and coding mechanisms, the way information was being coded, visual information. But what was happening, we're now into the sort of, well into the kind of 70s, plasticity was being discussed. Now, everybody knew that the brain is plastic in early life. So, depending on how you rear the child or an animal, the brain develops accordingly. The retina taught us that there is also plasticity, significant plasticity in the adult central nervous system. And again, that was a bit of a battle with editors and so on to use the term adult synaptic plasticity, which again has become a reality now because we know it happens as a function of sort of learning and memory and things like that. So that brought me, neuroscience to me, or brain, was like a biological universe. So what I learned from physics, I brought into neuroscience, and I could look at the way nerve cells were responding to stimuli or talking to each other in the way that, you know, I had learned about the physical entities in the physical universe doing as well. And of course, with plasticity, I got to learn a lot about systems' capacity for change, not just any old change. Change as a function of, as a functional requirement. So depending on whether the visual system was light adapted or dark adapted, these synaptic interactions would change. For example, that biphasic cell, red, green, depolarizing, hyperpolarizing, became monophasic under dark adapted conditions. And that kind of makes sense because we don't need color vision in the dark. So the cell kept in step. And that was my 25 years of neuroscience. And I loved it. And I did see eventually, when I did an MBA course on ion selective electrodes, and I inserted an electrode into barnacle muscle, this is giant muscle, and there it was, bang, you know, minus 70 resting potential and so on, correlating directly with where the electrode was. And then quickly moving across to cancer. Of course, cancer, when I came interested in cancer, that was pure curiosity. There was no emotional issue or any reason. I was just curious. And to me, that came across as a pathological universe. Cancer is incredibly complex. That's why it's so hard to deal with, to manage. But of course, you know, with the training from physics, and neuroscience, I felt kind of at home analyzing this complex world of tumors. Interestingly now, of course, these two fields have merged. Now there's a whole new field of cancer neuroscience. It turns out tumors are innervated, and tumors acquire neuronal properties as part of their behavior and so on. So this is what I meant earlier when I said, we're not just kidding when I say, you know, these are exciting times. Everything is gelling. And I'm sure you'll want to talk about, you know, how we're going to use bioelectricity in the clinic. But I think we are fast laying the grounds for exploiting bioelectricity in oncology as well.
Yeah, so maybe now is a good time to get into it a little bit. And I know you've already touched upon it, but perhaps if you can explain it more simply in terms of, you had a 1995 paper, which was your first paper, I think, on connecting ion channels and cancer. So how did you start connecting the two together? And then what would you say is the role of ion channels in cancer and just bioelectricity in cancer?
Right. So again, we wanted to adopt a comparative approach. We were convinced that ion channels would play a role in cancer. At that time, I asked two questions. This is how we were trained in physics. Always ask viable questions. Being able to ask a good question is much more difficult than finding the answer. If the question is good, there's a chance that you'll find an answer. If the question is bad, you'll be stuck. Anyway, the question was two questions. First, I'm embarrassed to say I was very naive, and that was, do cancer cells generate electrical signals? And this is now me thinking like an oncologist. You know, what's bioelectricity got to do with cancer? You know, this is cancer. This is not neuro. This is not muscle. It doesn't twitch. It doesn't, you know, buzz or something. And of course, that was indeed a very naive question because we now know every cell in the body has some kind of bioelectricity. By the way, even red blood cells, which do not have any genetic material, still have a small membrane potential. So it seems you can survive without genes, as it were, without a nucleus, but you still need some kind of bioelectricity. So, so the second question was, do these signals differ between strongly metastatic cells, cells that are capable of invasion and aggression and spreading around and ultimately killing you, versus those cancers that are benign. They sit there, they're a tumor, but they're not aggressive, they don't invade, and so on. And for that, I adopted a rat model of prostate cancer. It's a beautiful model, the so-called Dunning cell model. This is a set of prostate cancer cells with varying metastatic ability, all derived, by the way, from the same original tumor. So they're all isogenic. So we were really comparing, lemons with limes as opposed to lemons with oranges or something. And so when we did those experiments, immediately we discovered we were expecting something to do with calcium. Everything, calcium is everywhere, isn't it? So we thought we're going to get bogged down in some calcium signaling and try and sort that out. In fact, what we found was the expression of a voltage-gated sodium channel specifically in those strongly metastatic cells. By the way, why did we, there are lots of ion channels, as you know, lots of different types of ion channels. We focused on voltage-gated ion channels. Why? Because, like Mike, we knew what the membrane potential, what voltage can do to proteins, including ion channel proteins. And that minus 70 resting potential that we take for granted, if you express that as a voltage gradient, it's equivalent to something like 10 million volts over a meter. This is a huge force. And every protein in the cell membrane is subject to this force. So we thought we're going to go with voltage-gated ion channels. We'll deal with ligand-gated and mechanosensitive channels on a different day. The other thing that we did was to focus on carcinomas. Now, earlier we just been talking about gliomas. These are brain tumors, but we said to ourselves, look, brain is already full of ion channels. We go in there, look at some brain tumor, then we'll have God knows how difficult a job we're going to have sorting out the ion channels to do with the tumor versus those channels that are already there because it's brain. Whilst carcinomas being derived from epithelia, we thought would have a cleaner baseline, so we would have a better understanding of what was happening specifically in metastatic cells. And thirdly, we focused on metastasis because that is indeed, as we said before, a major cause of death from cancer. Those were the three prerequisites that we adopted. So when we looked at those cells, there it was. The sodium channel popped up, and then the non-metastatic or very weakly metastatic cells didn't have this. Now, a lot of people at that time said, this is an epiphenomenon. It's the first time I heard of that term, actually, epiphenomenon. So the beauty of working with ion channels is the availability of good pharmacology. I mean, neuroscience has come a long, long way. So, and we're spoiled for the ways in which we can analyze ion channels using drugs, using natural toxins. Of course, nowadays, we can do all the gene silencing and so on. So for sodium channels, of course, the beauty is tetrodotoxin, the pufferfish toxin, one of nature's most specific agents, deadly, by the way. So we use, it's quite difficult to get hold of TTX now because of it's associated with sort of terrorism and things. But we're so early on in the game that we could get it and use it. And there it was. This is a TTX sensitive current. You remove sodium, it disappears. You can bring it back and everything else. So and then came the point of proving that it's not an epiphenomenon. It actually contributes directly to the metastatic process. And for that, we did invasion assays. So we could put the cells into a sort of micropore filters, coat it with matrigel. Matrigel mimics the basement membrane in cells, put the cells on top, and the cells, the metastatic cells, digest the matrigel and then go from one side of the filter to the other. So we did all those things with and without TTX. And of course, we could show that we could block the invasiveness. Not black and white, not 100%, about 50%. This is the thing, cancer is not black and white. You're dealing with so percentages all the time. And so I sent that paper to Nature, came back saying, well, yeah, interesting. If you do something in vivo and if you look at this and the other, we'll look at it because we do think it's interesting. And I made an instant decision. I said, look, if I spent time doing all those things, I wouldn't last in the game. So we adopted a bottom-up approach. So the paper you just referred to came out in Pep's Letters, quite a respectable journal, and we built up from there. And as I keep saying, it's been absolutely sort of fascinating. So this phenomenon. The other thing I should note is whilst this voltage-gated sodium channel was being expressed in the strongly metastatic cells, their outward currents driven by voltage-gated potassium currents were reduced. So it looked like the cells were trying to become excitable. And of course, years later, using microelectrode arrays, we could show that these cells indeed generate bona fide action potentials. They are truly excited. So I call this the celex model. Celex stands for cellular excitability. So I hope you'll agree that, even my mother understood this when I explained to her that, to make cancer spread or to make cancer invasive, if it's going to kill you, what do? You make it excited, you make it excitable, get it out of control, make it twitch, make it disruptive, invade its surroundings and things like that. And how do you do that? You put some ion channels in there, turn it into a bit like an epileptic brain. So the whole thing, is twitching away. And then, yes, cancer is deadly, can be deadly and well-designed, difficult to handle, but there are certain aspects that are actually quite inefficient and rely on chance. So in that sort of scenario, as these cells are twitching under the influence of the things like the action potentials and so on, all they need to do is to, by chance, bump into a capillary, get into the circulation, and then could end up anywhere in the body. And talking of cancer's inefficiency, we know that of those cells, only one in about 10,000 survive. So it's not as if every cancer cell that goes into circulation will get to its destination. There's a lot of sort of inefficiency and redundancy along the way. But if it gets to the wrong place, it could be very, very problematic, of course. We know that.
That's fascinating. Did you see the voltage-gated sodium channel uptake in all types of cancer?
Yeah, now it's royal we, but we did in my lab, we did prostate, we did breast, we also did colon. There was a lovely paper that published on colon cancer from Georgetown University, showing that, first of all, they showed that the sodium channel is there. We call it sodium channel. I'm very, physicists are very precise. So strictly, it's voltage-gated sodium channel, because there are sodium channels that are non-voltage. But anyway, for simplicity's sake, we call it sodium channel today. So those guys, House et al. Showed that these human colon cancer cells express indeed the sodium channel. They also asked a very good penetrating question, saying, where does the sodium channel fit in this invasiveness or invasion mechanism or network? So what they did, they knocked out the sodium channel. They knew the subtype, NAV1.5. They knocked it out, and they looked at all the genes known to be associated with colon cancer invasiveness. And blow me, the sodium channel turned out to be the first domino in the network. So it is an early mechanism expressed early in the metastatic process, drives these other major mechanisms of invasiveness, including MAP kinase and Wnt signaling, calcium, proteases, and secretory mechanisms, and so on. And that really was also very, very important, because what everyone wants in cancer is early detection. Early detection saves lives. So the sodium channel is indeed an early event in the metastatic process. I mean, people have even said that expression of the sodium channel is necessary and sufficient for cancer cells to become invasive. Now, you know, this is a term that we used to use a lot in my physics days, necessary and sufficient. It's a very powerful way of giving a message. And this statement was made because those guys, this is the lab of Eric Bennett, they express sodium channels in a non-metastatic human prostate cancer cells, and it became invasive. So the only thing they did was to put in a sodium channel, and the cells became invasive. So it was necessary and sufficient. So this excitability phenomenon, or the way the sodium channels and the potassium channels combine in our vision, in our estimation, that's been seen in several cancers. But the sodium channel itself has been seen in almost every carcinoma looked at. The only exception in our hands has been pancreas. I mean, you know how difficult deadly pancreatic cancer is. I had a special PhD student working on this, and we really struggled to detect the sodium channel in pancreatic cells. We don't know why. In fact, we did form some ideas ultimately as to why not, but it's something we did not pursue. Maybe we'll go back to it one day. There's so much to do, for God's sake. There's so much to do. Just like, you know, Harvey Carton said to me at the time, Mustafa, just put all these bricks down the corridor and just blast your way through. Before the world woke up, you know, you will have done it all. I should say, going back to the 1995 paper, of course, as soon as we discovered the sodium channel and showed that it is a driver of invasiveness and metastasis, and then in vivo evidence came and so on, for it to become clinically viable, it had to be somehow different from the body's all other sodium channels. Now, sodium channels are a multi-gene family, and so they are in our excitable tissues as well as in some other tissues. So what was different about the cancer sodium channel that we could exploit clinically? So very early on, we looked at, of course, the answer was going to come from molecular biology. But very early on, we got into the molecular biology. I should say, you know, when I say we, one is people, the lives of people like mine are spoiled by our students. So I had a string of brilliant students, and Jimmy Dis had the idea, he had the insight that the sodium channel in breast by that time was an embryonic splice variant. It was developmentally regulated, and he knew which exon would be spliced and what that splicing, which part of the protein it would affect. We went there, sequenced it, and there it was. The sodium channel in breast and in colon is indeed an embryonic splice variant. And since then, we've gone on to making a monoclonal antibody for that, so we can target it. So, you know, these days, things like ADCs, antibody drug conjugates, are very, very favored as the new favored drugs in oncology, and our antibody has potential in that respect. So with the channel, we have one major clue for its clinical applicability. The other one was something that I had criticism for repeatedly. You know, at public meetings, people would again get up and say these things. So those two criticisms. One was, now Mike will know, cancer cells have very depolarized resting potentials. So most resting cells sit at minus 60, 65, 70. These tumor cells sit at minus 20, 25, 30 at the most. Now, under those circumstances, the sodium channels ought to be inactivated. But the fact remains that these channels are active because you put TTX on. We know exactly where TTX binds. It blocks the open channel. And because it blocks the open channel, for it to work, the channels must be open or opening all the time. So we kind of suspected that what we measure as the resting potential isn't actually a static phenomenon. That in fact, it could be somehow undergoing some kind of oscillation and so on. And recently, with my bioengineer colleagues at Imperial, we use voltage-sensitive dyes, avoiding dialyzing the cells with micropipettes and things. And we showed that indeed, cancer cell resting potential is very, very dynamic. And they start from minus 20, 30, whatever, and then they go through these, what I call hyperpolarizing voltage transients. And I think that's another chapter in this world, what those transients kind of do. Now, going back to the clinical potential applications and things, the other criticism I kept getting is, look, the sodium channel, the way it opens and closes over milliseconds, wouldn't bring in enough sodium to impact upon the cell. Now, everyone knows that cancer cells are loaded with sodium. And sodium MRIs have shown that tumors are loaded with sodium. So where is this sodium coming from? And this is something that if I may be allowed to be proud of, I sort of put together a bit like a jigsaw puzzle. And the answer came from the fact that under hypoxic conditions, these sodium channels do not open and close over a time scale of milliseconds. The channels actually stay open for seconds. And we call this the persistent current. And so that was, we immediately thought, well, damn it, everyone knows growing tumors are hypoxic because, once tumors grow, inside starts lacking in oxygen and so on. So why not? Maybe our channel undergoes this persistent current under hypoxic conditions. Do the electrophysiology, put the nitrogen in instead of oxygen, there it is, they develop a persistent current. And the beauty is there is a drug that blocks selectively the persistent current. This is ranolazine. It is actually an anti-angina drug because exactly the same phenomenon occurs in heart muscle during irregular heartbeat, arrhythmia or angina, something like that. When oxygen doesn't flow effectively and hypoxia develops, hypoxia, of course, is lack of oxygen. The sodium channel again stays open, sodium rushes in, and that disrupts calcium homeostasis and pH and so on, and you end up with heartburn. Ranolazine blocks that component selectively. And that is beautiful because that is what we wanted. We couldn't afford to block the whole sodium channel. I mean, it would give people heart attacks and, the nerves would seize up and muscles would freeze and everything. We needed to somehow tease apart either the sodium channel or some particular property of it so as to not affect the primary function, which is the transient current generating our heartbeat and action potentials and so on. And we've gone on to patenting that drug on the basis of secondary medical indication. Ranelagine, I wrote about these things, is now in preparation for clinical trials. Now, there is a bit of a twist to this because ranelagine is off patent. It's an old drug. I like old drugs. I don't know how Mike feels about these things. Mike, knows much more about electroceuticals than I do. But I like golden oldies that have stood the test of time. So ranelagine is a beauty in that regard. But it is off patent and hence subject to generic substitution and so on. And when I wrote about this first time, one of the referees picked this up. The paper was in a journal called Recent Patents in Cancer Research, in Cancer, something like that. And this reviewer said, look, the kind of drugs that Djamgoz is talking about are likely to be cheap. Is this going to be a hindrance to their commercial development? And, by that time, I had a company. I was talking to the big companies. And, again, Mike will know, sometimes we like to push reviewers' comments to one side and just skirt around them. And my first tendency was not to get into any discussion of it. And I thought, actually, it is an ethical issue. I must address this. And I wrote in the final concluding paragraph that the kind of drug that would work on sodium channels in cancer, in oncology, would be likely to be cheaper than existing drugs, cancer drugs, and we hope that this wouldn't be a hindrance to their adoption clinically. So there we are. And I got to a point in my life now where I'm much more, I really am truly motivated about getting to patients, in getting these non-toxic drugs that have significant potential, got to be tested, and hope that one of them will work. The other thing that we did, according to celex, yes, by blocking the sodium channel, you'll get an anti-metastatic effect, but we also brought in then the outward currents because we knew that by opening the potassium channels, we could generate inhibition in the cells. So it's a double-edged sword, not double-edged sword, double whammy. Anyway, so we combined ranelagine with a potassium channel opener called minoxidil, and we could get much better effects. That was in vitro, by the way. And so a lot of work remains to be done in vivo and so on. So we are well positioned. The concept has consolidated with our colleagues around the world are beavering away. Drugs are coming along, antibodies are coming along, companies are coming along, and I think it's just a matter of time before kinds of things that we are doing, some of which we started, become reality. And maybe we can mention, Mike, the fact that soon NCI, National Cancer Institute, will be running a kind of select workshop or seminar conference on cancer bioelectricity. So when guys at NCI, NIH pick these things up, then things become serious. So that's it. That's another step up. So where this brings me? And I did say this to Cancer Research UK when I got money from them. And I want to say something about funding. So we got money once we discovered or demonstrated the neonatal nature of the channel and wanted to make an antibody. I made an application to the technology arm of CRUK and got the money. And I said to those guys, I wish I started this work 20 years ago. So I could be 20 years ahead now. You know what they said? You wouldn't have got the money 20 years ago. The field had to be consolidated for us to believe you, as it were. And then you got the money and the rest. Now, talking of believing, this is one of my favorite experiences. So again, so Nature sent the paper back, but we're building up the story. And I was applying for funds and not getting any grants and things like that. But something did happen. I'll say two things, bits of experience in this regard. First was after we published the prostate story on those Dunning cells, I had a surgeon wanting to do his master of surgery in the lab. And of course, I was delighted. Medics are always fun to have in the lab, very, very interesting. So said to this guy, said, okay, you're a medic, you're going to work on human cells. So the guy would operate in the morning, rush to the lab with his bits of prostate tumor. I think he snipped a little bit from the normal side as well. And trying to do the electrophysiology turned out to be quite hard. We did get some results, but then we eventually moved on to cell lines and demonstrated again that sodium channel is there, block it, suppress the invasiveness and things, basically showing in humans what we had already shown on the rat cells. And this paper came out in the American Journal of Pathology. And at that time, Sunday Times, the major national weekend newspaper in the UK, telephones Imperial and asks, is anything interesting going on at college that we can make news of? So the college gives a list and they pick our research. And so pufferfish toxin, new hope for prostate cancer. I mean, look at how the journalists turn these things around. And because of the use of TTX and things like this is published in, I think the contact was made midweek. By the weekend, half-page article comes out in the Sunday Times. I was, what's the word I need? I was very nervous as to what these journalists would write. I was a young academic working with these high-powered oncologists. You know what they're like? I thought, my God, if they don't apportion the credit or they do something wrong, these are journalists, not scientists, and so on. In fact, this story was perfect. What I want to say, though, is Monday morning, the telephone in the lab went red hot. People telephoning to offer money to support the research. And I couldn't believe it. All those years I worked on the retina, nobody offered me anything like that. So after consultation with Imperial, we formed an internal research fund, which I eventually registered as an independent foundation. And that oversaw, that enabled us to cross some critical bridges in building up the evidence, those little funds. The other thing in relation to funding, that the charity still exists, by the way. It's still it's sort of semi-dormant, but I enjoyed doing charity work. I really think people who do philanthropy in the right way and associate with those sort of people are special, because you're doing something really purely for goodness, no expectations, no nothing. Now, the other experience was eventually I got a grant from the Medical Research Council for prostate cancer, which was surprising. Of course, as often happens, some of the people on the panel. So one of them said to me, he said, Mustafa, we haven't known what to do with you. We haven't known what to do with you. Meaning they could neither accept all the evidence that I was putting out, nor could they reject it, because, this was peer-reviewed publications and things. We haven't known what to do with it. And then, do you know why you got this grant? I said, why, Colin? He said, because you connected the sodium channel to epidermal growth factor, to a mainstream cancer mechanism. And then they understood. And that was a big lesson to me. Since then, I spent a lot of time associating functionally these channels with mainstream cancer mechanisms, meaning growth factors, hormones, tumor suppressors, mechanisms of apoptosis, and all these things. And that really helped, because people then started not believing anymore, but understanding. And that helped us a lot in really getting firming up our foot in the field, as it were. So, a whole world of experiences.
Mustafa, I have a question that this comes up a lot, and it's pretty heartbreaking. Like, I get a lot of emails from patients, from people who have cancer and so on, and they say, okay, I've been reading all your papers. I see it's all very exciting. What do I do? And so my question is, and of course the hope is that someday we have something to tell them other than, well, hang around, we'll get it eventually, right? So we want to have something. Is there anything that you're aware of that you can tell people that's actionable beyond talk to the best oncologist you can? Is there anything that you know of?
Mike, as you can imagine, I get that question all the time.
Of course.
And what I say is roughly what you would say, or say, listen, we will get there. Just be patient, just hang in there, and so on. But I've gone a bit further than that. In part through my charity, I still see a lot of cancer patients. Sadly, they often come to me when they're desperate, meaning they've been through the motions, nothing else to do, and then they think you and I have the magic wand. I have grown to appreciate integrative medicine, basically combining Western medication, chemotherapy, whatever, with natural agents, with lifestyle, and so on. And in this regard, what we were saying before, the fact that tumors turn out to be innervated now provide a physical mechanistic basis for the impact of brain on cancer. I'm not saying you can sit there and meditate and your cancer will disappear, but it's all these things help. So there's a paper in Nature, it's been some years now, where people induced, I think, 2 types of tumor. One was a melanoma, one was lung cancer. This is all in mice. And then they stimulated a part of the brain called ventral tegmental area. A very small part of the brain, about 10,000 neurons, something like that, uses the neurotransmitter dopamine, which is the so-called reward transmitter in the brain. And what happened was these tumors shrunk, or their growth slowed down. And this was evidence that brain activity does impact on tumorigenesis. And so I think, you can't, unfortunately, I can't offer them magical solutions, but we can give them hope. I think there are some practical things. Of course, the beauty of the sodium channel is, first thing I tell them is stop salt, cut out salt, sort of thing. So dietary things, pH, I'm a strong believer in alkalinity. Then I looked into this and published, because people, again, physiologists would say, yeah, but the body's pH regulatory system is so strong. It doesn't matter what you eat, what you do, pH will always be fixed. But it's not. If you do drink bicarbonate and so on, and go and measure the pH of the urine first thing in the morning, you do see that it's gone alkaline. So you can overcome some of these things. So I wrote a book, which I need to update actually, called Beat Cancer. It sells on Amazon as everything else. And the last figure in that book is a balance, which I drew with my own hands. That's how I see cancer and cancer patients managing their cancer or living with their cancer and its progression. So basically, in our lives, there are factors that add to the plus sign, make the cancer worse. It could be your diet, it could be smoking, it could be obesity, all those things. And then on the other side of the balance are the things that you can do to counteract that. So it could be green tea and alkalinity and vitamin D and things like that. And I think that's what I tell patients, Mike. Go to bed and ask yourself every night, what have I put on the minus side of this balance to keep it tilting away. But Aastha, this fits in with the title of the series, live longer world. In this instance, live longer with cancer, because we are saying at the moment I want to see cancer become a chronic disease that we can live with, and I think that's a first step. So if we can manage this balance, then it will give us hope to be able to live with cancer, certainly live longer with cancer, and so on. And so now the old wives' tales about positive thinking and this and the other now turn out to have a physical or mechanistic basis. So we ought to take that seriously. Yeah, I think cancer patients need management. For many years, we run a drop-in center for cancer patients in the north of England, where people would come and get this sort of advice, and we would actually hold their hands and often accompany them on appointments and things, and help them survive better. Of course, the sad thing is, and I used to teach medics. The day I got my first professorship, I said, I'm not going to teach medical students anymore. What medical students get on nutrition in 5-6 years of medical training is about half an hour or something like that. Whilst the old saying that we are what we eat is absolutely true. We are a biochemical engine. The difference between us and robots is our chemistry. And our chemistry originates from our diet. So it kind of, not that it makes sense, it gives people hope. And I think the other thing that gives people hope is that when we befriend them, when we sympathize or empathize with them, that also goes a long way, because it's not just that you are supporting the patient. Patient supporters also need support. So we are in a little society in which we can make some difference. Can we make all the difference? Not always, but some cancers are almost curable, manageable. Others not so much. But cancer, for sure now, is not a death sentence. We don't refer to it anymore as the big C. We use the word cancer quite easily, and so on. So let's hope that today that we can live with it. And I think we are. In my estimation, oncology, clinical oncology, is coming around to this concept of living with cancer. Let's try and turn cancer into a chronic condition. And there are lots of examples of this, like diabetes. We can live with diabetes. Of course, the big example is HIV and AIDS. Now HIV, the virus, can be controlled, and you can live with it without AIDS ever developing, sort of thing. So that's what from all the research and everything we have learned in this sort of world of bioelectricity, in which neuroscience and biophysics and oncology all mix, that from that mixture we can generate not just hope, but some practical reality.
It's a fabulous place to end on. Thank you so much, Mustafa. Yeah.
Thank you so much.
Thank you, guys.